Thank you very much for that introduction, and thanks to the organisers for inviting me. Very beautiful country. So what I'd like to do is really show you three fundamental studies we've done to not just to explore whether prebiotics have therapeutic potential in psychiatric disorders, but also whether they can be used to maintain brain health, to sustain a healthy brain or improve normal brain function. So I'll introduce prebiotics. I'll talk about an animal model, a mouse model of information related to anxiety. We looked at emotional processing in humans, and I'll go to neurobiology and cognition, which is really something we started with at the beginning of this interest in the microbiome. So what are prebiotics? Well, Ted's talked about, and as you know, living organisms in the gut, probiotics that can confer psychotropic effects. Prebiotics are actually dietary fibres, nutrients that feed the gut bacteria already residing in our gut. So our rationale for using prebiotics was to grow more species than the one or two species Ted gave you an example of, and through that perhaps confer even more psychotropic effects, because you're more likely to increase several psychotropic species. So what these are short chains of sugar chains. The most naturally occurring ones are fructooligosaccharides. There's several others. They're found in various foods there. But recently, a galacto-oligosaccharide prebiotic has been developed, and this is quite potent to grow bifidobacteria. It's quite bifidogenic. And this is the compound we've been working on mostly. It's commercially available as Bioimmuno or Bigos, I'll refer to it. And as you can see, fecal densities of bifidobacteria are quite high in GOS-fed animals. These are rats compared with FOS. Okay. So we thought we'll look into the effects of Bioimmuno, so we get more likely to get a hit. And as Ted mentioned, when these are broken down, they produce short-chain fatty acids. Now these acids they go into the bloodstream. Importantly, they actually release gut hormones and stimulate the immune system, and I'll come back to that. So, as you may know, the gut bacteria and the immune system are inextricably linked. Okay? The bacteria sit from birth really primed our immunity, increase in good bacteria tends to develop an anti-inflammatory response, and this is constant. This has been known for a while, and it's constantly being investigated, as you can see from here. What also has been looked at for quite a few years is the interaction between the brain and the immune system, and there's even genetic links showing that illnesses such as schizophrenia are associated with dysfunction of immune components like the MHA complex and so on. So this first started a few years ago, but there now has been a resurgence of this research, probably because of the interest in gut microbiome. So to study this at the preclinical level, a model was developed by Dancer a few years ago, and it showed, and as you can see here, there's more recent studies exploring this, that if you inject an animal a single acute injection of endotoxin and then just leave them alone, they develop a sickness behaviour, but 24 hours later, and even 48 hours later, they develop anxiety-like behaviour and depressive-like behaviour, depending on how many injections you give them. Okay? So we wanted to see, okay, there's a link between the gut microbiome, the immune system, and then anxiety. Will a prebiotic attenuate this? So, our question was, does Bigos, this is the potent prebiotic we're using at the moment, attenuate post-inflammatory anxiety in mice? So we injected mice with LPS. Some had prebiotic on board, they were fed a prebiotic, others were just given water and normal chow. And in fact, we did find that there was anxiolytic effect of the prebiotic. Our mice developed anxious behaviour, and the BGOS attenuated this, and some molecular markers, which I'll go into later, were changed as well. So what the actual technique was, it was a light dark box. So when an animal is anxious, it will just stay in the dark compartment of the box, and it won't come out. If it's less anxious, it will explore more. And so you can measure the time in the black area and the time in the light area, and you can also measure latency of how long it takes them to get out. So this is an example. So here you see that 24 hours after an LPS injection, mice that were just on a normal diet with water, they were spent less time in the light because they were more anxious. Okay? And those that were fed the BGOS, the prebiotic, actually it didn't affect them at all. So they were more ready to come out and explore the environment, the lit environment. Okay? So that indicated that the prebiotic had anxiolytic effect. And as I said, the latency was significant as well, so we had this significant interaction with the diet. Now, as I said, studies have shown that in an acute inflammatory response, obviously you've got the pro-inflammatory cytokines increasing in the brain, but you also get some neurotransmitter receptors. We're not quite sure what they're doing, whether it's a homeostatic response or they're key to the inflammatory response. But certainly the serotonin 2A receptor does seem to increase during a pro-inflammatory insult to the brain or a systemic injection of an endotoxin. So we wanted to look at this, and of course, as you know, serotonin is linked to anxious behaviour and depression. So that makes it more relevant to look at. And what we found, firstly, interleukin-1, the pro-inflammatory markers, is exactly the same as anxiety. In the brain, this is the frontal cortex, the prebiotic diet attenuated the increase in pro-inflammatory cytokines in the frontal cortex. We didn't see a change in the anti-inflammatory cytokines, which other groups have reported, just a suppression of pro-inflammatory response. Okay? In terms of serotonin, again, the 2A receptor increased in the cortex. In the presence of the prebiotic, it was attenuated. And there's another receptor, 5-HT1A, which was some antidepressants target. So that showed us, therefore, that the prebiotics do act through the immune system. Well, they grow the bifidobacteria. The bacteria interferes with the immune system or primes the immune system and suppresses the inflammatory response. So that's the immune mechanism we're exploring. The other thing we wanted to do is look at healthy human beings. Okay, so this is not a clinical population. We wanted to see, well, if it is anxiolytic and potentially pro-cognitive, can we just give healthy individuals this and we see an improvement in brain function? This is obviously important because you want to maintain brain health throughout aging. So what we did was we recruited 45 healthy volunteers, no history of psychiatric disorders, which is quite difficult in Oxford. And they were split in 3 groups, and they were given supplements of FOS, BGOS, maltodextrin, daily for 3 weeks. Okay, and we, as Ted mentioned with his study, we also measured salivary cortisol response. Okay, but this is the awakening cortisol response. As you know, cortisol is secreted as a stress response, endocrine stress response. But when you wake, there's a peak in it, and it goes down. We wanted to see if this peak changed. So then we looked at emotional test battery and subjective mood ratings. So you've got 2 things here. You've got subjective mood ratings were really testing if they felt anxious, happy, sad, the usual things you would expect to measure in the clinical population. But the important thing for us was to actually look at the emotional test battery. These are psychological mechanisms that underlie anxiety. So if you can affect those, then you may be able to tackle anxiety itself. If you can tackle the underlying psychological deficits that are there. So that's why we measured them. And what we found of course, or else I wouldn't be showing you, is that the prebiotic intake reduces the awakening cortisol response and alters emotional bias in healthy volunteers. So it did what we were hoping to do as we predicted from animal studies. And this is what this means. So here we have subjects on placebo, and this is their awakening. So when you wake up, it takes saliva samples, and there's a peak up to 30 minutes of waking, and then it levels up to goes down levels after 60 minutes. Okay? This was at the beginning of the trial, and this was at the end of the trial. So the blue line indicates this was measured 3 weeks after. Okay, the first set of samples. As you can see, the BGOS, which had anxiolytic effects, anti-inflammatory effects in mice, also reduced the neuroendocrine stress response, so reduced this. And interestingly, we were surprised about this. We did expect FOS to have some effect, because it does have some bifidogenic response effect on bacteria, but we actually saw that was quite flat. So we think that the difference is just by virtue of the bifidogenic properties of BGOS. Okay? These are areas under the curve, just to illustrate this quite significant, quite robust finding. Now, for the emotional processing, we didn't find, we had a test actually, we didn't find much. We did find a subtle change in attentional bias. Okay? So this is basically a computer task, and subjects are shown happy or neutral faces or sad faces. And the quicker their response to happy faces, the more positive their vigilance, the more positive their attention. As you can see in placebo, unfortunately most of us tend to look at negative images in life, but those who are on the BGOS tended to focus on more positive images. Okay? And so again, surprisingly, the FOS was actually worse. I'm not quite sure why. We would have expected something either like the control or maybe a trend like the BGOS. Okay? So this is emotional attention we're talking about. This is attentional vision. This is emotional cognition that we've measured. So nothing in terms of emotion. And of course, there was no changes in subjective ratings. So there was no changes. They didn't feel happy, didn't feel sad, didn't feel anxious. Okay? It didn't affect that. But that's okay, because we are actually finding that the underlying mechanisms of anxiety are changing. So if we can actually change this in a depressed group, for example, an anxious group, probably a few weeks later we will see a change in that. So if we tackle this first, we probably, if we fed a bit longer, we'll probably be able to see this a change in anxiety. So finally, going on to attention. Now, as I said, my interest has really been cognition, glutamine, and schizophrenia. When we look at cognition, when we looked at cognition in animals, we're looking at the N-methyldeaspartate receptor. This is hyperfunctioning in schizophrenia, and cognitive deficits in schizophrenia are thought to be a result of hyperfunction of this receptor. So we were quite interested to see whether the prebiotic actually affected the glutamate system per se. So again, we fed rats with prebiotics. The FOS again, we tried the FOS and GOS, this is at the beginning, and we did indeed find that not only did the prebiotic elevate brain-derived neurotrophic factor, which is a neurotrophic factor which probiotics have been shown to increase, with John Klein Dinan has actually shown that in their previous publications. So this was sort of our positive control. But we also found that the NMDA receptor was increased. But not only that, but the coagonist D-serine in the frontal cortex and the infrontal cortex. We also saw changes in the hippocampus. So these are quite significant. When we look at antipsychotic drugs, these are always as significant, and they're variable. So we're quite surprised that we even got a hit there. But certainly, what interested us was the change of the frontal cortex. We're interested in the executive function. And so to show this obviously might be a homeostatic response to the prebiotic, downstream functioning may not be occurring. So to test that, we did in vivo iontophoresis on prebiotic-fed animals. Okay? So in these experiments, animals fed on water or prebiotics are given NMDA in the prefrontal cortex. So it's iontophoresis. This is the full agonist of the receptor, and then you measure the response of the cells. Okay? And what we found is these are different currents which correlates to different concentrations of NMDA added. And as you can see, the response increases with concentration of NMDA. So we conclude then that these receptors that increase in a healthy animal's brain are actually functional. Okay? But again, they're functional in the frontal cortex. Do they actually affect behaviour? It could be a homeostatic response again, that's negating any downstream effects. So what we did, we then wanted to look at a behavioural correlate of NMDA function. And the best thing we could find was the attentional set shifting task. So this is a measure of medial prefrontal cortex function. Okay? It's a rodent equivalent of the Wisconsin card sort test. And what this does is basically the animals learn to discriminate between two different odours, and they have to find their food reward according to which odour is the relevant cue. Okay? And then you change the odour, so for example, if this odour indicated, that was the odour where the food reward was, and then you swap it, the animal has to learn that it's no longer that's the relevant cue, but that is. Now this is the intra-dimensional, when you change within one dimensional, i.e. odour. What we then do is we swap it, we change the rules and go to an extra-dimensional. So here the animal has to learn that it's the texture of the digging medium that's important, that will tell it that where the food reward is, not the odour. And this shift from an intra-dimensional to an extra-dimensional discrimination is the intra-dimensional, extra-dimensional shift, which is a measure of cognitive flexibility. Okay? And what we found was, of course, that the shift from, these are normal animals, this is usual, animals find it difficult to, they learn an intra-dimensional set, they know that odour is important, but then you change the rules, and it takes them quite a few trials to learn the new rule. When you actually put them on BGOS, they learn quite quickly, so they're not cognitively rigid. The cognitive flexibility has increased. Okay? So that showed that from the change of the NMDA receptor down to behaviour, and importantly, these are healthy animals, so that was quite surprising. So BGOS supplements healthy rats demonstrate greater cognitive flexibility. So of course the implications of this is that we can now go to patient groups such as schizophrenia and any other patient groups where there's profound cognitive deficits and just see if these do make changes. But what about the mechanisms then? I'll just briefly finish off with this. As I said at the beginning, the short-chain fatty acids which are generated from prebiotics, the gut bacteria generate these in the presence or absence of prebiotics anyway from our food, from the fibres. But you get more short-chain fatty acids with prebiotics because there are more fibres for them to digest. So there is a link with short-chain fatty acids immunity, and as I said, gut hormones, GLP-1 and PYY, peptide, tyrosine, and these are key in the release. These are released after food intake, and these are satiety hormones. So whenever we eat a meal, these are immediately released, and it causes satiety. So our hypothesis was that these are generated by the bacteria. They generate short-chain fatty acids, release these, and then one or both go to the brain and cause changes in the NMDA receptor. Both of them are brain penetrant. So, simple experiment, we just got some rats, we injected rats with PYY or GLP. This PYY is the most predominant soluble form, circulating form, and we did indeed find that there's an increase in cortical NR1 subunits and also the coagonist D series. So we think with prebiotics at least, the increase in the gut hormones is really influencing the NMDA receptors, possibly over changes in the immune system, but we've yet to prove that. Okay. So, to summarise, central effects of prebiotics, so they don't have an effect themselves, they're not absorbed, they transit through the colon, and the bacteria feed on them, and then the majority of them are bifidogenic. Prebiotic BGOS is mainly bifidogenic. We've shown it has anti-inflammatory, anxiolytic effects, but not on its own. There had to be an inflammation response, injection of endotoxin. We didn't find any basal effect. And BGOS improves emotional attention, so cognitive, emotional cognition in healthy volunteers, and improves cognitive flexibility in animals, possibly through PYY. GLP we didn't see have an effect. Okay? But I think, I know Ted is a great advocate of the single species or multi-species probiotic, but I do think that these are most likely to be adjunctive treatments. I don't think they are powerful enough to tackle severe depression, cognitive deficit, schizophrenia. I think they will help the brain respond more to current medication, but we've yet to prove that. And finally, just to get a taste of what we're doing, we recently found, we're going, as Ted also said, to neurodevelopment. This is relevant to us, schizophrenia and cognition. And we found that BGOS also increased glutamate subunits and synaptic proteins in rat pups when they were fed for a couple of weeks postnatally, from day 3 to day 21. And then when we let them grow to early adolescence, these changes were sustained. But certainly, we do advocate theory that changing your microbiome early in life does influence the development of your brain through whatever mechanism, immunity, short-chain fatty acids, gut hormones, and so on. So what's left for me to say is, obviously I don't do any of this work. It's all down to my colleagues in Reading, Leicester, and I have started collaborations in London as well. And thank you very much for your attention.